Welcome to today's lesson on the stages of protein synthesis, where we will explore the fascinating processes of transcription and translation. The goal of today's lesson is to allow you to describe these stages clearly and understand how they contribute to the larger biological functions within cells. In our roadmap for today, we will break down protein synthesis into its two main stages: transcription, where DNA is used to create messenger RNA, mRNA, and translation, where the mRNA is decoded to assemble proteins. By the end of this lesson, you should be able to describe each of these stages in detail and understand their significance. So far, you have learned about the structure of DNA and the concept of the genome. You understand that DNA is made up of nucleotides and is organized into chromosomes, which contain our genes. This foundational knowledge about DNA's structure and function provides the essential groundwork for understanding how genetic information is translated into functional proteins. Understanding the stages of protein synthesis is a crucial advancement in our biological understanding, because it explains how genetic information is expressed in cells, leading to the formation of the proteins that carry out virtually all cellular functions. This knowledge is fundamental for fields ranging from genetics to medicine and biotechnology. Before we dive into the process of protein synthesis, let's quickly recap what you already know about DNA and genes. A gene is a segment of DNA with a specific sequence of bases. This sequence encodes the instructions for creating a sequence of amino acids. Every 3 bases in a gene correspond to one amino acid. The particular order of these bases determines the type and order of amino acids in a polypeptide chain, which ultimately forms a protein. Now, let's look at the key roles proteins play in the body. For example, enzymes are proteins that act as biological catalysts, speeding up chemical reactions. Another example is maltase, which breaks down maltose. Hormones are proteins that carry messages throughout the body. For example, testosterone is crucial for male reproductive development and secondary sexual characteristics. Structural proteins provide support and strength, such as collagen, which strengthens connective tissues like ligaments and cartilage. There are many other types of proteins with various functions in the body. With this background, let's explore how these proteins are made through the processes of transcription and translation. Transcription occurs within the nucleus of a cell. Here are the stages. First, the DNA double helix unwinds to expose the gene that needs to be copied. An enzyme called RNA polymerase binds to a non-coding region of the DNA just before the start of the gene. RNA polymerase moves along the gene, creating a complementary mRNA, messenger RNA copy of the DNA sequence. Once the mRNA is formed, it leaves the nucleus through a nuclear pore to reach the cytoplasm. In RNA, the base uracil replaces thymine. So A pairs with U, and C pairs with G. Translation takes place in the cytoplasm at the ribosome. Here's how it works. The mRNA attaches to a ribosome, which reads the mRNA sequence in triplets called codons. Each codon on the mRNA specifies a particular amino acid. Transfer RNA, tRNA molecules in the cytoplasm carry specific amino acids to the ribosome. Each tRNA has an anticodon, which pairs with a complementary mRNA codon. The ribosome forms peptide bonds between the amino acids brought by tRNA, creating a growing polypeptide chain. This process continues until a stop codon is reached by the ribosome, signaling the completion of the protein. By understanding both transcription and translation, you now have a complete picture of how the intricate blueprint stored in your DNA is converted into functional proteins that perform a myriad of essential functions in your body. This knowledge is foundational for delving deeper into the field of genetics and biotechnology. Let's work through a question to consolidate our knowledge. A section of DNA had the base sequence A, T, C, G, A. What would be the base sequence of the section of mRNA that has been transcribed from this DNA? For questions like these, it is important to remember that the complementary base to C is G, and T is A. In mRNA, the base uracil is used instead of thymine, so A is complementary to U. Therefore, the complementary mRNA sequence to the DNA section would be U, A, G, C, U. In today's lesson, we explored protein synthesis, focusing on transcription and translation within the context of DNA's role in inheritance and evolution. We learned that DNA contains instructions for making proteins, which determine an organism's traits. During transcription, a gene's DNA sequence is transcribed into messenger RNA, mRNA, in the nucleus. This mRNA then travels to the ribosome in the cytoplasm. In translation, the mRNA is read by the ribosome in codons, with each codon specifying an amino acid. Transfer RNA, tRNA, brings the correct amino acids to form a polypeptide chain, which folds into a functional protein. This lesson emphasized how genetic information is transferred from DNA to proteins, highlighting DNA's crucial role in inheritance and diversity.